Hopefully this works. Hello. I'm just making this video because I thought that this would be more convenient as well, I guess it's a check that we've gone over the study and so that we all understand what's going on right now. So I'm just gonna very quickly go over and summarize the study. Hopefully I don't take more than 10-15 minutes. I'm just gonna go over what all these sources are. There's 5 of them right now as I determined what would be useful. And then if you guys have any questions, you can always ask in the group chat. And if you want a specific role for the group, as in if you want to do background or methods or whatever, just tell me, because I'm going to ask you guys anyway. So yeah. All right, so let's start with this study. So we have nuclear receptors and we have nuclear receptor co-repressors. NCOR1 is a co-repressor responsible for adaptive transcriptional changes, and it's used as scaffolding protein that interacts with a lot of other proteins. Those proteins we don't have to list. If we do, it's pretty much just going to be fluff because no one's going to understand it. But it's used in, they all participate in a very complex signaling pathway. Generally, NCOR1 is supposed to prevent or really suppress cardiac hypertrophy and tumor development. And one of the problems that researchers have had one of the problems that researchers have had testing what would happen if they took out this gene is that the germline mice that inherit the gene for the double negative on the NCOR1 end up dying in the embryonic stage, and they usually don't go beyond that. So a few liberties were taken when producing these mice to ensure their survival and to test the conditions. Now, in the methods, we're probably not going to spend more than 2 slides on this. Anything else, it's going to be a little too complicated, but we're basically going to divide it into how we generate the mice without NCOR1 and how we quantifiably measured the physiology of the wild or control mice versus the mutant and modified mice. Their DNA being modified, that is. So originally, we start off with a genomic DNA sequence of the lab rats. And we use a method called floxing to excise NCOR1. There's a pretty good image of it here, just to give you an idea. So floxing is when we use a polymerase chain reaction, which multiplies the gene surrounding NCOR by millions and billions of copies. And that action pretty much pushes the NCOR1 gene off of the DNA strand. You can sort of see it here. They're targeting exon 11, which you could assume is NCOR1. And when they expose it to the high fidelity polymerase chain reaction, it's essentially pushed out, it's knocked out. Now, after that, they use electroporation to stimulate gentamicin resistant, that's the G418, embryonic stem cells to allow the modified DNA to enter. Electroporation is a fancy term for electrically stimulating the cell membrane to create holes that allows DNA to enter. And then they allowed the homologous recombination of DNA inside these cells to fully integrate the absence of NCOR1. Now, the next step that I wrote here is more for trivia, I guess. So This is just to make sure that there really is no NCOR1 in the gene and to confirm the point mutation, the deletion. They did a digest on the DNA, they electrophorized it, and they put it on a transfer membrane to ensure that it really was deleted. And that can be seen here. Anyway, this part isn't really too important. I'm actually going to strike this through now. But if it needs to be explained, we'll at least have the information here to do that. But anyway, after it was confirmed that the DNA samples had no NCOR1, the stem cells were injected into mouse embryos, the blastocysts, that were then implanted into surrogate mothers, pseudo-pregnant mice, which produced the first batch of modified children. And over the course of time, these mice were bred with other generations of mice, including ones with genes for human skeletal actin, that's for human muscle fibers, to create the final batch of mice. My rationale was that they wanted to make sure that the lack of NCOR1 actually affected human skeletal muscle. So in order to not actually have to test it on humans, why don't you just give a mouse human muscle cells? Simple as that. So that's on generating them. And for actually measuring the physiology of the mice, we described the conditions that they're kept in, the diet that they were fed, there's two of them, controlled diet, pretty average, and the high fat diet, which is essentially a keto diet, if you know what that is. And then we took blood samples, tissue samples, we performed calorimetry, we did oral glucose tolerance tests and parental glucose tolerance tests. We did enzymatic assays to basically take a basic metabolic panel. We measured their blood pressure with this computerized tail cuff system. We measured the performance of their heart using echocardiography and the muscle density of the heart. Now, when we get down to the actual results, there are three things that we're looking at: the muscle mass, the exercise performance, and the oxidative capacity. These two, the muscle mass and the exercise performance, are the main ones stated in our hypothesis. And this last one, oxidative capacity, is what's responsible for the former two. So one thing to note first, the NCOR1 gene was selectively excised and reduced in muscles in the legs and the pericardium of the mice. So as stated here, it was significantly decreased in the soleus and gastrocnemius, which is in the calves, the quadriceps, and modestly reduced in the heart muscle of the altered mice, but no other tissues were manipulated. It was in the results that the density of, the mass of the calf muscles were higher in the mutants than in the controls, but that in the gastrocnemius was not statistically significant. And they didn't really specify why, and we don't really need to either, because it's not really important. So you can actually see over here in figure 2C, that's 2, that's C, comparing the controlled mice, that's SKM++, to the mutants, that's SKM negative, that there is no actual visual distinction that you can make between the muscles themselves. However, those without NCOR 1 had muscles that were seemingly more intensely red. Now, they also noted that there was no real weight changes or changes in food intake between the control and the mutants, whether they were on different diets. So the main drawback that we get from here is this relative increase. And of course, over here, we have the conditions, which explains why we're mostly testing these muscles. Now, second, there's a focus on the exercise performance, and this is where it gets more interesting. So what this first line is saying about the total oxygen consumption being increased, the decreased respiratory exchange ratio, is what I interpret as the respiratory system of the mutant mouse being more efficient. If you find that I'm wrong, please correct me. Now, there's also the second figure that shows, I believe it's 1D. This is 1, that's D. The black being the mutants, the white being the controls, that the mice were much more cold resistant than the unmodified mice, as they were able to maintain their heart rates at colder temperatures. They actually tested this until it went down to 4 degrees Celsius. Just as a reference, that's about 36 degrees Fahrenheit, pretty cold. And you can already see that at 34 degrees Celsius, their heart rate is already decreasing, while the modified mice are steady. The most obvious result between the difference of the modified and the control mice is, I suppose it was done using some kind of small treadmill or a running wheel, because they had these mice run, they measured their speed and the distance that they ran. So here in figures 3A to C, you can see that the mutant mice not only ran farther, but they generally ran faster. And as stated over here, this is without any real differences in heart rate, blood pressure, cardiac morphology, or function than their controlled counterparts, meaning that they were able to run faster and longer while maintaining the same heart rate as the control mice, which I think is somewhat impressive. Lastly, for the oxidative capacity, this one is more histologically focused, so we look closer at the cell types. Now, it was noted that, and you can see in all of these diagrams, that the NCOR 1 negative mice, the modified mice, had a change in the muscle fiber type that they expressed. Now, the first thing is there was less connective tissue present overall in the modified mice. And you can sort of see that here. You can see, I guess this is from the bird's eye view from up top, and this is from the side, that there are less, in between the actual muscle cells, there is less space. They're grouped together more closely. And you can really see that in the second diagram. Now, I'm sorry if you noticed the pause. The second thing that we notice is, as I stated before, with the switch to more oxidative fibers, that according to staining with succinate dehydrogenase and the cytochrome C oxidase, that there is more staining, at least by ratio, between the controlled and the modified mice. And this is also shown in this chart next to it. And this is supported by both kinds of staining. And this is meant to signify the greater mitochondrial activity between these fibers. So as opposed to the fibers in the control mice that prefer glycolysis, these fibers in the modified mice prefer the mitochondrial activity. And the last thing to really note here is that the diameter of each of the muscle fibers in the modified mice's muscles is larger than that of the control mice. Don't be confused by the chart. This is for the gastrocnemius. So you see here, this is meant to represent that there are more control mice in this category, the 30 to 35 micrometer per muscle fiber. And there are more modified mice here in the 45 to 50 category, meaning that they have larger muscle diameter. And this is even more significant in the soleus, and both of these are in the calf. So that's just a summary on pretty much the entire study. We've gone over the results, the methods, and the a little bit of the introduction. Aside from that, the discussion is really what we make of it. I think that the results sort of speak for themselves, that this, you know, the deletion of NCOR1 genes does lead to what we expected in the hypothesis. So we can accept it, but you know, as I spoke to Hilmi today, we should also, in either our discussion or the conclusion, make note of the detrimental effects of the NCOR1 deletion. We must remember that this is important to stop cardiac hypertrophy and tumor progression. And if we don't have this co-repressor, we may see uncontrolled growth in these areas. Now, just for the sake of time, I am going to put labels for these resources, what they're good for. Actually, honestly, I might not even need to, because the titles kind of say everything, but I'll still look into them and I'll label them. If you have any questions, just let me know. Otherwise, you guys just let me know what roles you want, or I will choose them for you. Goodbye.